RBCs carry oxygen. They are normally under very high oxidative stress. But RBCs don't have any nucleus. So how do they produce this enzyme? And what happens in G6PD deficiency? In this video, we'll look at what happens in G6PD deficiency, what are the clinical features, a little bit about the genetics, and in the end, a comparison between hereditary spherocytosis and G6PD deficiency. I'm Nabeel, and this is Pathocyte. Let's first look at the basics. G6PD enzyme produces NADPH. This is used by glutathione to produce reduced glutathione. And this reduced glutathione prevents oxidative damage. Now let's look at what happens in G6PD deficiency. Here, although G6PD is produced, the produced enzyme is not stable enough. So most of the enzyme cannot be used. Because of this, NADPH is reduced. So the availability of reduced glutathione is lowered. So there is no one to prevent oxidative damage. This is the basis of G6PD deficiency. Now let's look at the pathogenesis. Oxidant stress inside the RBCs lead to denaturation of hemoglobin. This denatured hemoglobin goes and sticks onto the RBC membrane. These are called Haines bodies. Now, depending upon the severity of damage, in G6PD deficiency, we can get intravascular or extravascular hemolysis. If the damage is severe, RBCs burst inside the blood vessels, causing intravascular hemolysis. Now, if the damage is partial, RBCs lose their membrane, and just like in hereditary spherocytosis, they become small, dense spherocytes. These spherocytes are not flexible, and just like in hereditary spherocytosis, when they pass through splenic cords, they are eaten up by splenic macrophages, leading to extravascular hemolysis. Another interesting thing which occurs is when these RBCs with Haines bodies travel through the splenic cords, these Haines bodies are eaten up by the splenic macrophages. And the RBCs end up having a very unique shape. They look like somebody has taken a small bite out of them, just like the symbol of apple. So these cells are called bite cells. So till now, we saw that there is not enough glutathione in G6PD deficiency. So oxidative stress inside RBCs is very high and hemoglobin denaturation occurs, which leads to the production of Haines bodies. If this leads to severe damage of RBCs, intravascular hemolysis occurs. But if the damage is partial, most of the RBC membrane is lost and spherocytes are formed, which are eaten up by splenic macrophages, leading to extravascular hemolysis. We also saw that sometimes Haines bodies are eaten up by splenic macrophages, leading to the formation of unique bite cells. Now let's briefly look at the genetics. G6PD deficiency is an X-linked recessive disorder. So, males are more commonly affected. Most of the females are mosaics. G6PD deficiency is most common in the Mediterranean region, North and Central Africa, and the Middle East. This is common for most of these hemoglobinopathies and inherited enzyme disorders and membrane disorders of RBCs. It is thought that this has something to do with survival ability from falciparum malaria. survival of the fittest. No wonder inherited RBC disorders are so common. Now let's figure out what are the clinical features. Episodic hemolysis is a feature of G6PD deficiency. But why does it occur? Hemolysis in G6PD occurs almost always after a trigger. This trigger can be any sort of oxidative stress, most commonly infections, certain drugs, and fava beans. These can produce oxidative metabolites. In G6PD deficiency, older RBCs have very less amount of functional G6PD, but younger RBCs still have decent amount of G6PD enzyme. So whenever there is this trigger, all the older RBCs are destroyed, but the younger RBCs survive. So the hemolysis is always self-limited. That means once the older RBCs are all destroyed, hemolysis stops, and then recovery occurs in the form of reticulocytosis because of marrow response to anemia. Now clinically, when there is a trigger, patients can have acute hemolysis leading to fatigue and jaundice, as most of the hemoglobin which is released is converted to bilirubin. Now if there is a massive intravascular hemolysis, these patients can have hemoglobinuria. And this free hemoglobin in the kidney can lead to acute kidney injury leading to back pain. To summarize, we saw that G6PD deficiency leads to reduced oxidative stress management in RBCs, which can lead to severe damage of RBCs causing intravascular hemolysis or partial damage leading to extravascular hemolysis. And then we saw that G6PD deficiency is an X-linked recessive disorder, and clinically patients usually have episodic hemolysis due to a trigger, which can be drugs, infections, or fava beans, or any other oxidative stress, which can clinically present as fatigue, jaundice, hemoglobinuria, back pain, etc. Now let's look briefly at the peripheral blood findings in G6PD deficiency. In the peripheral blood, you get to see these unique cells called bite cells. Then, if you do a supravital staining, you can see Haynes bodies. In routine Romanowski stains like Leishman, these Haynes bodies do not take up stain. So, in routine staining, we get to see Haynes bodies like this. So these cells are called blister cells, where the RBCs look like they have a blister, which is nothing but Haynes bodies which have not taken up the stain. You may see spherocytes in the smear, and if there is an acute hemolysis in the recovery period, you get to see reticulocytes, which have these blue dots because of retained RNA. Now let's look at some important differences between G6PD deficiency and hereditary spherocytosis. G6PD deficiency is an enzyme defect, while hereditary spherocytosis is a membrane defect. Both are inherited. G6PD deficiency leads to episodic hemolysis, while in hereditary spherocytosis, you get to see chronic hemolysis. In hereditary spherocytosis, splenectomy helps, but in G6PD deficiency, splenectomy is not helpful. If you have come till now, you have just understood the most common inherited enzyme defects in human beings. To summarize, G6PD deficiency is an X-linked recessive disorder. It is pretty common around the Mediterranean region and North and Central African regions. Here, RBCs are destroyed because of oxidative stress, which is normally managed by G6PD, but here in these patients, the enzyme is defective. This can lead to intravascular or extravascular hemolysis. And clinically, patients present with self-limited episodic hemolysis. In the peripheral smear, you get to see distinct cells like bite cells, blister cells, etc. In the next video, we'll look at thalassemia. Think at the cell level.